afternoon, everybody. I'm going to talk about Stabilitech Biopharma and how we can redefine the way we make and take vaccines. Stabilitech has three platform technologies. The first is a platform technology that completely removes the cold chain for viruses wherever they be used, whether it's vaccines or also in the biotech space for CAR T cells or for oncolytic viruses. It's obviously a very important thing for the cold chain for vaccines where there's huge amounts of wastage. The second platform technology is antigen sparing, where we can reduce down by 1/20th the amount of antigen required to elicit an equivalent immune response. That obviously has benefits for the manufacturing outputs in terms of increasing the numbers of doses that you can get from a single manufacturing run, but it also offers the interesting potential to include more viruses, more antigens in a single vaccine, such that you can increase the breadth of immunity for a patient. And the final platform that we have is our oral delivery platform. It's a viral vector, a non-replicating viral vector that's delivered to the GI tract and can elicit an immune response just from an oral capsule or tablet. Oral delivery technologies significantly reduce the costs in terms of manufacturing and delivering at the actual site, and also speed up the manufacturing and research and development times. It also removes needles from the whole paradigm of delivering a vaccine, something that costs healthcare workers a lot of injuries. Our pipeline includes three products that I'll talk about today. The first deals with the influenza virus market. There's a constant problem with flu strain selection mismatch that costs patients a lot of infections, lives and economies, an awful lot of money. We can deal with that by vastly increasing the number of influenza strains we can include in a single vaccine. That's utilising our antigen sparing technology. The second product is our oral Zika virus vaccine that's completely thermally stable, can be shipped anywhere in the world, and will deal with the issue that 50% of the people who actually have the potential to suffer from the Zika virus will never actually see that vaccine because of thermal stability losses throughout the cold chain. And the final product is a genital herpes vaccine, again using the oral delivery vector technology, thermally stable, etc. Patients suffer an awful lot of morbidity with genital herpes. 80% suffer more than 2 episodes in their first year. 60% suffer more than 4 episodes. Huge numbers on a worldwide basis are infected and suffer from new infections every year. A brief history of Stabilitech Biopharma. Started in 2002. Had initial investment, and following on from there, we gained funding from both sides of the Atlantic, started to develop our internal pipeline, have partnered and formed collaborations with big pharma, veterinary healthcare companies, and also started to push on our own internal pipeline further. And most recently, we've signed an agreement with an oncolytic virus company, Efficacious Biopharma, over in Japan, to thermally stabilise their virus. Delving into the seasonal influenza market, flu strain selection is a continual problem. They get the flu strain selection wrong 60% of the time. That's primarily because they can only select one strain to represent, for example, H1N1, one strain to represent H3N2, and so on and so forth. And because of that, the vaccine effectiveness never rises above 45 to 60%, even on a well-matched year, and on a poorly matched year, that will nosedive down to as low as 3%. That has an immediate impact on the local population in terms of the numbers of infections and the cost to the economy in both direct and indirect healthcare costs. Flu is not going away. This will be a continual story every year until it's solved. It's an annual epidemic. The market is huge. It's forecast to grow to 4.3 billion by 2025. Over 300 million are vaccinated, and the market has already displayed a propensity to accept the principle of more vaccine strains gives better protection because it readily adopted the quadrivalent vaccine, the 4-strain vaccine, over the 3-strain vaccine. If we can move from 4 strains to 10 strains, we will completely turn the tables on the flu strain selection match issue, and may also increase the overall vaccine efficacy because you will better represent the number of circulating strains in the environment on a given season. To have a little bit of a look at our OmniFlu vaccine, this is our flu vaccine. It utilises our antigen sparing, so we can deliver 1/20th of the dose required for each virus strain. It's a multi-strain vaccine, so it's a super seasonal vaccine. There's 10 strains are included. It utilises whole inactivated virus, so we see an authentic immune response. Both the humoral and T-cell responses are engaged there. It's adjuvanted to ensure that immune-compromised populations also benefit from this vaccine, and we're completely agnostic to whether it's cell-based production or egg-based production. Just to have a quick look at some of the preclinical efficacy that we've gained. As you can see, this is a 15-valent vaccine, so above what we need to do. It was administered to naive mouse, IM, and as you can see, after the prime boost dosing rate regimen, it was cross-reactive to all the strains presented in the vaccine, but also strains that weren't selected in the vaccine, and most interestingly, to pandemic strains as well. So we're really engaging some interesting epitopes by our downstream processing that we carry out to see this antigen sparing. Just to have a look at the vaccine market in general, and this is delving into our oral delivery platform. The vaccine market is already big. It's going to grow to 77 odd billion by 2024, but there are key unmet needs. The development timescales can be long. The vaccine delivery costs in terms of administering via a needle and the cold chain, etc., can be large, in some cases larger than the actual vaccine itself, and the wastage is still huge. What we've done is develop a plug-and-play platform. The little part of the red piece of DNA can be plugged in and out for any viral antigen, and that gives us many vaccine development opportunities. And because you become very familiar with your platform, you get speed and flexibility from that. You also get some manufacturing know-how and regulatory dossier information that can be transferred from one program to the next. But you can only reuse a vector if you can go through the oral delivery administration route, because otherwise you'll get anti-vector immunity. So by delivering the viral vector to the GI tract, you can do repeat administration, and there's all the additional benefits of oral delivery. Reduced cost of administration, we see systemic immunity developed, and also there's patient preference. But you can only deliver to the GI tract if you have thermal stability. The stomach is a challenging environment, both in terms of temperature and acidity, and also you want to have that thermal stability for worldwide vaccination campaigns and the ability not to see huge amounts of vaccine wastage. So does this work? Here's some data, non-human primate data from our oral Zika virus vaccine. As you can see, there's 3 groups. The first group in blue had previous exposure to the whole Zika virus and had acquired natural immunity, so they had a previous infection, they had natural immunity. The second group in the burnt orange received a placebo vaccine, and the third group in the green received our candidate vaccine. And as you can see, after an aggressive intravenous challenge with a live virus, the placebo group suffered a huge amount of viremia, whilst the vaccine candidate group showed a strong immune response equivalent to that of the group that had natural immunity. And this is a product that was shipped outside of the UK without a cold chain and was still efficacious on use despite having sat on a tarmac in a country trying to gain access for a period of time. We will now be utilising this platform for our genital herpes programme. It's a huge market, as I mentioned, over 120 million infected in the major markets throughout the world. There's no vaccine available. it's genuinely a $1 billion opportunity, and this program is now in preclinical studies. In terms of our timelines to progression, as you can see, by the end of 2023, we aim to have phase 2A readout data for both of these programs. And we're seeking up to 30 million to enable that to happen. There's $1 billion upsides for both of these programs. They're both in huge markets. It's worth pointing out that AuraPro-Zika is funded by the UK government to be CGMP ready, so we will have material ready for a first-in-man study, hopefully by the end of next year. And obviously there's many other vaccine candidates have an opportunity to be developed off that platform. Just to give you a quick overview of our board and scientific advisory board. And there we go. Hopefully I was on time.